For 150 years, we got it wrong.
Every memory you cherish, every fact you've learned, every face you recognize. We thought neurons controlled it all. We were wrong. In January 2025, scientists discovered something extraordinary. Your memories aren't made by neurons. They're controlled by a cell we dismissed as worthless. Using one of the most dangerous molecules in your body. Madrid, 1887. A struggling Spanish physician named Santiago Ramón y Cajal sits in his attic laboratory, peering through a microscope he bought with his military discharge pay. What he sees will change everything. Using a silver staining technique, Cajal discovers something revolutionary. Neurons are separate cells, not a continuous web, individual units. He calls it the neuron doctrine. In 1906, he wins the Nobel Prize, and neuroscience becomes neuron science. For over a century, neurons became celebrities, the stars of the brain. Every textbook, every documentary, every animation showed neurons as the heroes of thought and memory. Billions of dollars poured into studying them, millions of research papers, entire careers built on understanding how neurons fire, connect, and remember. But there was a problem. Neurons make up about 10% of your brain. The other 90%? For decades, we called them glia, from the Greek word for glue. Support cells, scaffolding, nutritional suppliers, janitors cleaning up after the important neurons did their work. We didn't give them names. We barely studied them. They were the stagehands while neurons took every bow. But here's what we missed. There are more glial cells in your brain than there are stars in the Milky Way galaxy. About 85 billion of them. And one type in particular, shaped like a star, turned out to be hiding the biggest secret in neuroscience. Astrocytes. From the Greek astron, star. Each astrocyte extends hundreds of delicate arms, reaching toward neurons, wrapping around blood vessels, touching up to 2 million synapses at once. They're everywhere, watching everything. If they're just support cells, why do they need to touch so many synapses? Why does your brain dedicate so much real estate to something that's supposedly just glue? And why, when scientists finally looked closely in the 2000s, did they discover that astrocytes respond to neural activity with waves of calcium signaling? That doesn't sound like glue. That sounds like communication. In January 2025, researchers in Paris made a discovery that changes everything we thought we knew about memory. And it all comes down to a molecule you probably have under your bathroom sink: hydrogen peroxide. H₂O₂. 2 hydrogen atoms, 2 oxygen atoms. You use it to bleach your hair, clean wounds, and whiten teeth. In your cells? It's usually a disaster. Hydrogen peroxide is what's called a reactive oxygen species, ROS. Here's the problem. That extra oxygen atom, it wants to react with everything. Your DNA, your proteins, your cell membranes. This is oxidative stress, the same process that makes iron rust, apples brown, and, scientists believe, contributes to aging. In the brain, oxidative stress has been linked to Parkinson's, ALS, and especially Alzheimer's disease. Plaques of damaged proteins accumulating over decades. This is why we're told to eat antioxidants, blueberries, green tea, supplements. Fight the oxidative damage. Eliminate those reactive oxygen species. So here's the question that kept researchers awake at night. Why do our cells deliberately make hydrogen peroxide? Not as waste, not by accident, on purpose.
Your cells have dedicated enzymes, molecular machines, whose entire job is to manufacture hydrogen peroxide. They're called NADPH oxidases. Evolution doesn't waste energy building molecular factories for toxins. Unless, what if hydrogen peroxide isn't poison at all? What if, in the right dose, at the right place, at the right time, it's a signal? Think of it this way. Fire can destroy a city, but a single match can light a candle, send a message, guide someone home in the darkness. The difference is control, dosage, location, timing. Starting in the early 2000s, researchers discovered that cells use tiny, controlled bursts of hydrogen peroxide as a signaling molecule. It tells cells to divide, to defend against infection, to respond to insulin. But in the brain, we had no idea what it was doing. Until now. Paris, 2024. A team of researchers led by Yasmine Raba and Thomas Préat at ESPCI Paris are studying memory in fruit flies. Yes, fruit flies. Before you question this, fruit flies have been the workhorse of neuroscience for a century. They're genetically tractable, their brains are simple enough to map completely, and their basic memory mechanisms are surprisingly similar to ours. The team trains flies to associate specific odors with negative experiences. The flies form memories. But here's what's interesting. They use genetic tools to manipulate specific cell types. When they shut down NADPH oxidase, the enzyme that makes hydrogen peroxide, specifically in astrocytes, the flies couldn't form long-term memories. Short-term memory? Fine. The flies remembered for a few minutes. But ask them to remember something for days? Nothing. The memory just vanished. But why? What was the hydrogen peroxide doing? The team spent months imaging live brains, watching molecules move, tracking signals. And what they discovered is the most elegant memory system evolution has ever designed. It all starts with a neuron trying to remember something. Imagine you're learning something new. A face, a fact, a phone number. Your neurons fire. Electrical signals race across synapses. But here's what we missed. Neurons aren't talking just to other neurons. They're talking to astrocytes. The neuron releases acetylcholine, a neurotransmitter. Golden molecular packets drift across the synaptic gap. Some reach the next neuron, but some reach the astrocyte wrapped around that synapse. The astrocyte has receptors, specifically NACHR alpha 7, nicotinic acetylcholine receptors. Yes, nicotinic, like nicotine. Same receptor family. This is why smoking affects your brain, but that's another story. When acetylcholine binds, the receptor opens, and the astrocyte wakes up. Calcium ions flood in. Not everywhere at once. This isn't chaos. The calcium flows through the astrocyte's arms like a wave through fiber optics. Precise, directional, controlled. This is the astrocyte's internal language. Calcium waves encode information about what's happening at each synapse it touches. Remember, a single astrocyte touches 2 million synapses. It's receiving reports from all of them, all the time. When calcium levels surge in one region, the astrocyte knows. This synapse is important. This memory matters. The calcium wave reaches NADPH oxidase. and the molecular factory turns on. NADPH oxidase starts pumping out superoxide, O₂ with an extra electron, even more reactive than hydrogen peroxide, even more dangerous. The astrocyte releases these superoxide molecules outside its membrane, into the tiny space between cells. And here's where evolution reveals its genius. The astrocyte also secretes an enzyme called SOD3, superoxide dismutase 3. Its job? Transform superoxide into hydrogen peroxide. It's elegant. Take two superoxide molecules, add water, and you get hydrogen peroxide plus regular oxygen. Safer, more stable, but still reactive enough to carry a message. But SOD3 needs something: copper ions. And where does the copper come from? The neurons themselves provide it, through a protein called amyloid precursor protein, APP, on the neuron's surface. Remember that name, amyloid precursor protein. We'll come back to it. So now we have hydrogen peroxide molecules floating in the space between astrocyte and neuron. These molecules don't wait around. They're too reactive. They slip through the neuron's membrane, small enough to pass through like ghosts. Inside the neuron, the hydrogen peroxide molecules are recognized, not as damage, as a signal. They trigger what's called a redox signaling cascade, changes in the cell's chemistry that turn on specific genes, genes that make proteins, structural proteins that strengthen synapses, receptors that make the neuron more sensitive to future signals. The synapse physically changes, gets bigger, stronger, more stable. This is long-term potentiation, the molecular basis of memory. And it doesn't happen to just one synapse. The pattern spreads. Every neuron that fired during that experience, every connection that was active when you learned that fact, saw that face, heard that song, gets strengthened. The memory locks in. Not because the neurons did it alone, because an astrocyte, using one of the most dangerous molecules in biology, told them to. Pause the video. Take a moment. Every memory you have, your first kiss, your graduation, your child's birth, what you had for breakfast this morning, all of it required this exact process. Astrocytes, using controlled bursts of hydrogen peroxide, permanently changed your neurons. You are the sum of all your astrocytes' work. Now here's where the story gets darker. Alzheimer's disease affects 6.9 million Americans. Worldwide, 55 million people. And every year, that number grows. We've known for decades that Alzheimer's involves plaques, clumps of a protein called amyloid-β accumulating in the brain. We've spent billions trying to clear those plaques. Most treatments failed. We were fighting the symptom, not the cause. Remember amyloid precursor protein, APP, the protein that supplies copper for SOD3? In Alzheimer's, APP gets cut incorrectly. The fragments, amyloid-β peptides, accumulate. But here's what the Paris team discovered. Before the plaques even form, these amyloid-β peptides do something devastating. They bind to the astrocyte's acetylcholine receptor, the same receptor that receives the signal from neurons. They block it. Watch what happens. In a healthy brain, neuron fires, acetylcholine released, astrocyte receptor activated, calcium wave, NADPH oxidase turns on, superoxide generated, SOD3 converts to hydrogen peroxide, neuron receives signal, memory forms. In early Alzheimer's, neuron fires, acetylcholine released, but the receptor is blocked by amyloid-β. No calcium wave, no NADPH oxidase activation, no hydrogen peroxide. The neuron tries to form a memory. It strengthens temporarily, but without the astrocyte's hydrogen peroxide signal, the memory fades, like writing in sand before a tide.
And here's the cruel part. Amyloid precursor protein, the source of both the copper that helps make hydrogen peroxide and the amyloid-β that blocks its production. The same protein. Two outcomes. Processed correctly, it helps build memories. Processed incorrectly, it prevents them. As amyloid-β accumulates, more astrocytes get blocked. More memories fail to form. More synapses weaken. More neurons die. The memory centers of the brain, hippocampus, entorhinal cortex, shrink. Not because the neurons were directly destroyed, but because they stopped receiving the signals they needed to maintain themselves. The conductor fell silent, and the orchestra forgot how to play. But here's why this discovery matters. For the first time, we understand the mechanism. If amyloid-β blocks the astrocyte's acetylcholine receptor, what if we could unblock it, or bypass it, or enhance the astrocyte's response? What if we could boost SOD3 activity, or safely deliver hydrogen peroxide signals artificially? These are questions, not answers. Drug development takes decades. The brain is staggeringly complex. But for the first time, we're not just trying to clear plaques and hoping memory improves. We understand the signaling cascade. We know where it breaks, and we can start designing molecules to fix it. So the next time you remember something, anything, thank a neuron, sure, but thank an astrocyte first. That star-shaped cell we called glue for a century, it's not supporting your memories. It's creating them, using one of the most dangerous molecules in your body, in doses so precise, so perfectly timed, that a memory lasting a lifetime forms in milliseconds. Your brain is stranger and more beautiful than we ever imagined, and we're only beginning to understand it. This is Cosmic Canvas, where we reveal the hidden architects of your mind. Until next time, remember something precious today, and thank an astrocyte.